Good morning. My name is Deepa Bajetha. Today I am going to discuss about bioenergetics. List of contents. First is introduction, concept of free energy, principle of bioenergetics, important state functions for study of biochemical reactions in which I will explain gives free energy, enthalpy, entropy, relationship between change in free energy, enthalpy, and entropy, redox potential, high energy compounds, classification of high energy compounds, adenosine triphosphate, ATP, structure and functions. First is bioenergetics. The term bioenergetics is made up of two words. First is bio and second is energetics. Bio means life or living and energetics means study of energy. So basically bioenergetics is the study of energy change in the biological reactions, or we can say that bioenergetics is the field of biochemistry concerned with the transformation and use of energy by living cells. The main goal of bioenergetics is to describe how the living organisms acquire this energy, how this energy transform and utilize in order to perform any biological work or task. So the study of metabolic pathway is thus essential to bioenergetics. Two type of chemical reactions performed in any organisms for their metabolisms. First one is catabolic reaction and second one is anabolic reaction. In catabolic reactions involve the breakdown of chemical molecules, and in anabolic reactions involves the synthesis of compounds. So adenosine triphosphate ATP is the main energy currency for the organisms. The goal of metabolic and catabolic process are first is to synthesize ATP, to synthesize adenosine triphosphate, and this adenosine triphosphate is synthesized from the environment, or to break down ATP into adenosine diphosphate in which two phosphates are groups are present and inorganic phosphate IP by utilizing it in biological process. In the cell, the ratio of ATP adenosine triphosphate or adenosine diphosphate concentration is known as the energy charge of the cells. A cell can use this energy charge to relay information about the cellular needs. If there is more ATP, if there is more ATP, more adenosine triphosphate than the adenosine diphosphate available, the cell can use this more ATP to do work. But in case if we have less ADP, if we have more ADP, more adenosine diphosphate and less ATP, then cell must synthesize ATP via oxidative phosphorylation. Next is concept of free energy. Every living cell and organism must perform work to stay alive, to grow, and to reproduce. The energy process in living organisms are defined by the basic law of thermodynamics. The energy actually available to do work is known as free energy. The free energy is the energy available in a system to do useful work and is different from the total energy change of any chemical reactions. Change in the free energy. Here we explain the free energy in the form of delta G, and delta is used to indicate such a change. So change in free energy are valuable in the predicting the feasibility of chemical reactions. The reactions can occur spontaneously if they are flow with the decrease in free energy. During a chemical reactions, heat may be released or absorbed. Cells require a source of free energy. Cells are isothermal system. Isothermal system means having constant temperature and constant pressure. There are two types of cells. Photosynthetic cells. Photosynthetic cells are quite diverse and indicate cells found in green plants, phytoplankton, and cyanobacteria. And these cells acquire free energy from absorbed solar radiations. And heterotropic cells, these are an organism that eats other plants or animals from for energy and nutrients, and they acquire free energy from the nutrient molecules. Cell transfer this from free energy into adenosine triphosphate and other rich compounds to provide energy for biological work. Next is principle of bioenergetics. Biological energy transformations obey the laws of thermodynamics. So two types of thermodynamics, first law of thermodynamics and second law of thermodynamics. According to first law of thermodynamics is based on the principle of conservation of energy. For any physical or chemical change, the total amount of energy in any closed system remains constant. Energy may change form or it may transported from one region to another. Only formation can be changed or it may be transferred from one place or one region to another, but it cannot be created or destroyed. So second law of thermodynamics is universe tends toward increasing disorder. Heat is transferred from one body to another body only if the two bodies are at different temperatures. In all natural processes, the entropy of the universe increase unless energy requiring process counteract it. The important state function for the study of biological system are there are different type of state functions like enthalpy, energy, entropy. The first one is the Gibbs free energy denoted with G, capital G, which is equal to the total amount of energy capable of doing work during a constant temperature and constant pressure. The delta G, change in Gibbs free energy. If the delta G is negative, then the process is spontaneously. And the term exergonic. What is exergonic here? Exergonic process is one which there is positive flow of energy from the system to the surrounding. The positive flow of energy from the system to the surroundings. And second one is if delta is positive, then the process is non-spontaneous and terms endergonic. And endergonic process that requires energy to be driven, absorbing energy in the form of work. If delta G, if delta G is equal to 0, then the process has reached equilibrium. Next important state functions is H, that is enthalpy, which is the heat content of the system. Enthalpy is the amount of heat energy transferred or heat absorbed or emitted in a chemical process under constant pressure. If delta H is negative, the process produces heat and is termed exothermic. When delta is positive, the process absorbs heat and is termed as endothermic. The important state functions for the study of biological system are the entropy, denoted as capital S. It is a quantitative expression of the degree of randomness or disorder of the system is called entropy. Degree of randomness or disorder of the system. Entropy measures the amount of heat dispersed or transferred during a chemical process. When delta S means change in entropy is positive, then the disorder of the system has increased. When delta S is negative, change in entropy is negative, then the disorder of the system has decreased. So next is relationship between the change in free energy, enthalpy and entropy. So here is equation delta G is equal to delta H minus T delta S. Delta G is change in free energy, delta is H is change in enthalpy, T is temperature, delta S is change in entropy, and the conditions of biological systems are constant temperature and pressure. Under such condition, the relationship between the change in free energy, enthalpy, and entropy can be described by the expression where capital T is the temperature of the system in Kelvin. So this equation can be defined as the enthalpy is the sum total of all the energies, whereas entropy is the measure of change in enthalpy and temperature. T represents the absolute temperature in Kelvin. So next is redox potential. Redox potential of any system is the electron transfer potential, which is denoted as E naught. The oxidation and reduction. Oxidation means removal of electron and reduction means gain of electron. So the oxidation and reduction potential may be defined as the quantitative expression of the tendency that a compound has give or receive electrons. So the redox potential of the system may be calculated from this following equation. Here E naught is standard potential, redox potential in volts at 298 degree Kelvin. N is number of electron transferred and E is redox potential. So E is equal to E naught plus 0.0592 upon N log concentration of reducing agent upon concentration of oxidizing agent. In bioenergetics, redox potential is the redox of in bioenergetics, redox potential is the ratio of NAD positive to NADH positive plus H positive. It describes the ability of NAD positive for metabolism. When a substance exists both in the reduced state and in the oxidized state, the pair is called redox potential. High energy compounds. Certain compounds are encountered in the biological system which on hydrolysis yields energy. The term high energy compounds or energy rich compounds are usually applied to substance which possess sufficient free energy to liberate at least 7 calorie per mole at pH 7.0. Certain other compounds which liberates less than 7.0 calorie per mole lower than ATP hydrolysis to ADP adenosine diphosphate plus inorganic phosphates are referred to as low energy compounds. If any compounds contain or liberate less than 7.0 calorie per mole, then those compounds are called low energy compounds. So all the high energy compounds when hydrolyzed liberate more energy than that of ATP, adenosine triphosphate. Most of the high energy compounds contain phosphate group. Hence, they are also called high energy phosphates, except acetyl coenzyme A. Here is classification of high energy compounds. First class is pyrophosphates. An example is adenosine triphosphate, pyrophosphate, in which two phosphate groups are attached. Second one is acyl phosphate. Here one phosphate group is attached with this functional group 1,3-biphosphoglycerate, carbamyl phosphate, acetyl phosphate. Third one is enol phosphate, in which one phosphate group is attached with this group, and the example is phosphoenol pyruvate. Thiol esters, here as thiol is attached with this group, example acetyl coenzyme A, acyl coenzyme A. Guanidophosphates, example is phosphocreatinine and phosphoarginine. So next is adenosine triphosphate. What is ATP? So ATP is a multifunctional nucleotide used in the cell as a coenzyme. It is often called the molecular unit of currency of intracellular energy transfer. ATP transports chemical energy within the cells for the metabolism. It is produced by phosphorylation, photophosphorylation, and cellular respiration, and used by enzymes and structural proteins in many cellular processes. So one molecule of ATP contains three phosphate groups, and it is produced by ATP synthase from inorganic phosphate and adenosine diphosphate or adenosine monophosphate. When we convert adenosine triphosphate, first it is converted into adenosine diphosphate, then further it is converted into adenosine monophosphate. So the three main functions of adenosine triphosphate in cellular functions are: first, it transporting organic substances such as sodium, calcium, potassium through the cell membrane, synthesizing chemical compounds such as protein and cholesterol, supplying energy from mechanical work such as muscle and contraction. So it is the structure of ATP. The structure of this molecule consists of purine base adenine attached to the first position carbon atom of the pentose sugar ribose. This is the first carbon atom. Here attached nitrogen base adenine. Three phosphate groups are attached at the carbon 5, C1, C2, C3, C4, C5. At C5 carbon, three phosphate groups are attached as the pentose sugar. It is the addition and the removal of three phosphate groups that interconvert adenosine triphosphate. Here three phosphates are attached, adenosine diphosphate, two phosphates are attached, and adenosine monophase, here one phosphates are attached. Thank you.